Right, in today's lecture we're going to be talking about dihybrid crosses. And a dihybrid cross is a breeding experiment which tracks the inheritance of not just one, as we would in a monohybrid cross, but two heritable traits. And this is very important for allowing us to understand Mendels' law of independent assortment, so law of mixing. And essentially, each pair of alleles separates independently during gamete formation, and it leaves us with a formula of 2 to the power n, where n is the number of heterozygotes, so the number of heterozygous individuals or heterozygous offspring. So, here is a question. How many gametes will be produced with the following allele arrangements? Remembering that the formula is 2 to the power of n, where n is the number of heterozygous individuals. Remember, not homozygous. So that's important. So try and work it out. How many gametes will be produced with this allele arrangement? This one and this one. Obviously, number 1 is far easier than number 2 and number 3. But all you've got to remember is to calculate how many gametes will be produced. You've just got to use the formula, 2 to the power of n. How many heterozygous individuals will be produced when we cross those allele arrangements? Here are the answers. If we have a look at number 1, there are 2 heterozygous individuals, 2 in the middle. So, in order to work out how many gametes, we insert 2 in place of n, and we get 4. How many heterozygous individuals in the second one? Well, 3, which means 2 to the power of 3 is 8. For the last one, you can work it out, but there can only be 6, or there can be 6 heterozygous individuals, which gives us 64 gametes, which is quite something. So, let's have a closer look at a dihybrid cross. Remember, dihybrid crosses can be asked based on the P's that we've done, or any other allele characteristics or alleles that we might want to cross. So you could get this in tests and exams, and you should practice how to do these. But if we take our old chestnut seed shape and now incorporate seed color into the assessment, we will see that we have round shaped leaves, seeds, and wrinkled seeds, and then we have yellow and green seed color. Okay, so this is what the genotypes will look like. Those are the number of gametes for each one. Now, knowing the number of gametes, because this is what you have to do first, is to calculate number of gametes, okay, using the formula that was just provided on the previous slide. Then you can do your cross, which is the typical Punnett square. But the Punnett square, remember, needs to be done on the gametes, because these traits come from each parent, right? So these are all possible gamete combinations. And now we're going to do our Punnett square. Right, so there it is. On the gametes, remember it has to be on the gametes, and there we have for the one parent, and there we have for the second parent. Cool, right. What does it look like? Here it is, and we can fill it in. And there we go. Those are the results of a dihybrid cross. Typical Punnett square approach, which is very easy to do once you have determined the number of gametes. Remember the formula, 2 to the power of n, where N is the number of possible heterozygotes. So, when we're having a look at the phenotypic ratio of a dihybrid cross, we get the following results. 9 round yellow seeds, 3 round green seeds, 3 wrinkled yellow seeds, and 1 wrinkled green seed. So, phenotypic ratio of 9 to 3 to 3 to 1. That's what it would actually look like. If we did a dihybrid cross, that's what the seeds would look like. Some of them would be wrinkled and yellow, others would be round and yellow, others would be round and green, and only one, remember, because it's the recessives, 2 recessives, would be green and wrinkled, okay, giving us that combination, that phenotypic ratio that you see on the screen there, 9 to 3 to 3 to 1. So, here is a nice summary of Mendels' laws. This is a very important table for you to use when you are revising. Remember, we have the laws on the left-hand side, so it's the law of dominance, the law of segregation, and the law of independent assortment, best shown by using a dihybrid cross. So what I've got there is I've got the parent cross and what the resulting offspring would be. Tall to short individuals, all of them would be all of them would be tall because of the law of dominance. If we have segregation, where we have both 2 tall parents that are heterozygous, we'll end up with our characteristic 3:1 ratio. 75% will be tall, 25% will be short. And then independent assortment will give us what we have just had a look at. Round versus green, round versus green seeds, and we get the 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16, based on how the gametes mix before they are fertilized.